So yeah, we call this infrastructure AGRASIM, agricultural simulator. It's now a brand new research facility, although we are building it already since a couple of years, but everyone who's familiar with public building activities knows that it can take much longer than you initially anticipate. So this is an infrastructure which we will use for simulating the effects of climate change on agroecosystems. And the motivation is that we want to simulate future crop production in vivo and in silico, meaning that we, on the one hand, have very detailed experiments in controlled environment at the mesocosm level with all possible measurements, basically real-time monitoring, and using this data for then improving our numerical models, and then later use our supercomputing capacities at Jülich to model all these processes, crop growth, nutrient and water cycling, and gas emissions at also larger scales, and integrated in our terrestrial system modeling platform. So the, let's say, more detailed aims of this infrastructure is to study the interactive effects of elevated CO2 warming and drought together on crop performance, plant carbon allocation patterns, water nutrient use efficiency, soil carbon cycling, storage, but also mobilization by mineralization, greenhouse gas and reactive end gas emissions, bleaching then of dissolved organic carbon, nitrate, phosphate, sulfate, and other nutrients or other elements, basically. Feedback then of the agroecosystem to the atmosphere and the hydrosphere by means of greenhouse gas emissions, and also nutrient leaching, and also resilience of the system to environmental stressors. We have, I mean, in the recent past, there are many publications on especially the interaction of different climate change factors, which require then also in experiments to combine these factors. So, for example, of drought and elevated CO2, or increasing atmospheric demand on water and carbon fluxes, then combination or interaction of drought and temperature, also subsoil warming, and the role of warming of soil at greater depth on carbon and nutrient cycling. Then also, demand for improved models, so that we can more or less or more realistically simulate then future climatic conditions, or the effect of future climatic conditions on crop growth and ecosystem performance. And there's also, has recently been perspective paper in Nature Climate Change by colleagues from Belgium that already have a kind of ecotron system where they also run such climate change experiments. I will introduce that a bit in more detail later. So how does AGRASIM relate to PhenoRob? We have, let's say, similar research questions, but that partially match PhenoRob questions, but also go beyond them. So how does global change affect plant performance, yield, water, nutrient use efficiency, and greenhouse gas and other gas emissions? And which agricultural management options are the most suitable ones for mitigating the negative effects of global change, but also for optimizing resource use efficiency, so water and nutrient use efficiency. And a key approach is that we do experiments under realistic conditions, but also future climatic conditions that goes then also beyond PhenoRob. We monitor all processes in all compartments, in the air, in the atmosphere, in the plants, and in the soil, and have a specific focus on water, nutrient use efficiency, and carbon storage. So what do we have available? It's a research facility with 6 controlled units consisting of a climate chamber each, a plant chamber, and a lysimeter. So the plant chamber is within the climate chamber, so specifically then protected against, let's say, ambient conditions. And the plant chamber itself is then attached gas tight to a lysimeter. The lysimeters have a surface area of 1 square meter and a depth of 1.50 meters, so to allow more or less realistic rooting depths for crops. They will contain an intact soil core, which we take in the field with a heavy machinery, basically, and then bring these intact soil cores back to our facility. There we have a transport vehicle that can transport these lysimeters, which will weigh between 3 and 4 tons. Then we have process control unit that supplies these plant chambers where all conditions are really monitored and controlled. So with clean air, with humidified air, CO2 at different concentration levels, and then we have artificial light here, an LED array with 8 channels. And then, I will describe these plant chambers in a bit more detail. So we can then control each unit independently with a unique set of climate data. So we can run, let's say, 6 different scenarios if we want. We can then vary the air composition with respect to CO2, relative humidity, and ozone in large ranges. So the air and soil temperature range is between -10 to +40 or even beyond. +40, relative humidity between 0 and 90%. I mean, not 100 because we want to avoid condensation. I will come to that a bit later. Light intensity between 0 and up to 2,500 micromole per square meter per second. Photosynthetically active radiation, which is, the 2,500 is equivalent to the sun intensity at noon at the equator. So that was our aim, that we can also simulate sunlight at realistic intensity. A spectral range from UVA to far red, CO2 up to 2,500 ppm, ozone up to 100 ppb, and we can irrigate with a precision of 0.1 millimeter of rain, which is then equivalent to 100 milliliters per square meter. We monitor, of course, air temperature and humidity, soil temperature and moisture, water content, but also soil water tension. The lysimeters are weighed with a precision of 100 gram. So also to have this water balance precisely on the lysimeters. At the lysimeter scale, gas exchange, or a whole range of different gases is monitored online. Also isotopes specifically, partly, not all of those gases, but water, CO2, and N2O will be also monitored isotope specifically. Then we will be able to measure soil isotope profiles with gas permeable tubing at 6 different depths. Also measure leaf fluorescence, and in the future also with ground penetrating radar for selected lysimeters. By this we can get also a highly resolved soil moisture distribution in the soil core. So we can also, if we want, apply stable isotopes, trace element fluxes by this, apply, for example, or do pulse labeling with 13 CO2, or apply isotopically labeled water from the top by irrigation, but also from the bottom by pumping water to the lower boundary of the lysimeter. So I will now describe in a bit more detail the concept of the plant chamber. So the ultimate goal was to avoid condensation. Why? Because whenever you get condensation in the chamber, it highly affects negatively the gas measurement, because gases dissolve in water, particularly CO2, but also the other gases. Like ammonia, they have a high solubility in water, but also methane and N2O get dissolved in water. Whenever you get condensation, it's fresh, pure water that is then dissolving gases very eagerly. But it will also change the isotopic composition of the water vapor, but also of the other gases, because these condensation and dissolution processes cause isotopic fractionation, which will then make our isotope-specific measurements useless, basically. So therefore, we spend a lot of time when we develop this chamber to avoid condensation at any time, all conditions, even at freezing temperatures. And therefore, the first step was to place the plant chamber, which has all the conditions we implement on the plants, in a climate chamber to protect it from outside temperature fluctuations. Then we wanted to have a high-performance multi-channel LED lighting system and a gas-tight connection to the lysimeters, but that will not disturb the weighing of the lysimeters. That was also the challenge. So we then tested 3 possible technical solutions for the cooling concept of the plant chamber. The first concept was a ceiling fan. So this was only numerically simulated by our colleagues from the Central Engineering Department of Forschungszentrum Jülich. So the first concept had a ceiling fan and the cooling of the side walls, but the resulting temperature distribution showed that we got a very, let's say, heterogeneous temperature, would have got a very heterogeneous temperature distribution with too high temperatures in some parts of the chamber, because the cooling capacity of this concept was not sufficient. So then we checked the second concept with a cooling plate here at the side walls, with a fan at the side walls. But also here for this concept, the cooling capacity was not sufficient. Then we tested the third concept, cooling elements in so-called cooling towers here, which you can see in this thermal simulation, where we have 2 cooling elements here, and a fan each, and then counter clockwise the airstream. So 1 cooling tower with an upward stream of air, and distributing this into the chamber through those outlets here. And then another cooling tower with a downward flux of air, and by this creating also an airstream, quite homogeneous airstream through the chamber. And we found then that this cooling system was sufficient, or the capacity of this cooling concept was sufficient, but required a high air and water flow, airflow through the chamber and water flow through the cooling elements. But finally, we then decided to go for that concept. This now is the simulation of the air velocities in the chamber with this cooling concept with the 2 fans, also showing that we have quite homogeneous distribution of airstream here, no dead volumes basically, which would then also be prone to condensation. So ultimately, or finally, it looks like this. So we have the cooling unit here in that, or heat exchanger in that box here, and the fan below, and then the glass tower here that is then distributing the air to the chamber, or on the other side, sucking the air back in from the chamber. We used glass because we wanted to have as little as possible reactive surfaces. All the metal surfaces here are coated with silicon oxide, so that we have minimized then also the metal surfaces that could potentially react with gases and change then the composition of the gases emitted by the plants or the soil. So that's how the concept now looks like. We have at the bottom the lysimeter that is connected gas tight but flexibly to the plant chamber, which then has, of course, a hole in its bottom. All the side walls and the bottom and the top are made of glass. Especially the top is made of a very highly transparent and UV permeable sealing glass. And the side walls are resistant to 900 kilogram of load, pressure load per pane. And all ceilings are made with Viton gaskets pressing, because this is a material that is also not, or that is inert, basically not chemically reactive. I've already shown these glass towers here with fused on outlets, and everything is made of glass, especially, I mean, the sidewalls and the doors and also the ceiling at the bottom. And all metal parts are coated with silicon oxide, as already mentioned, to reduce the reactivity. That's now the arrangement. We have 6 units, so 3 and 3, 3 in the front, 3 in the back, and then all the process control on top of it, and then clean air supply unit outside, and also the cooling units outside. And then here again, the climate chamber, plant chamber, and the lysimeter in yellow here in the basement. So that's how it looks or looked like when we constructed the facility. You see here that we asked the company to drill holes in our floor so that we then can bring in the lysimeters from the bottom, from the basement, and hang them into the ceiling basically, so that they are reachable from the basement. So step by step, we then had to build a steel construction on the one hand on top of all the climate chambers for the control, and what you cannot see here in the basement to support the ceiling, because now we have 6 holes in the ceiling, and each hole has to carry a weight of at least 3 tons for the lysimeters and another 3 tons for climate and plant chamber, and so there needs to be a support below in the basement, otherwise everything would collapse. And then step by step, the climate chambers were built, and now this is the final stage. Basically, you see all the process control units here on top of it, and then 3 climate chambers here and 3 more at the back. And now everything is basically ready to be used. This is now the first plant chamber that was constructed and built by our central engineering department. And you see also it's partially mirrored. The idea was that when the light is entering from the top, that not a substantial part gets lost through the sidewalls, through the glass of the sidewalls, so that we basically channel the light downwards to the bottom. And this concept works also quite well. And now you can see it, transport of the first chamber to our hall here. And then that's the final place now in the climate chamber. So also the climate chambers, of course, have to have this hole in the bottom, so that there's a direct connection then to the lysimeter, which is reaching into the basement. So this was now the first test with the unit number 1, which we used as a prototype before we really constructed all other 5 units and 5 chambers, plant chambers. And we saw that the temperature, light, and relative humidity control worked very well. I mean, some fluctuations here we still had. I go into more details just right afterwards. But we were quite satisfied already with the temperature control and the relative humidity and light control, which we have here in this 21-hour data set. And black are the respective set points, and red is the actual temperature, blue the actual humidity, and green the light intensity, actual light intensity. We then refined this control even more, so that we now can control the temperature and the humidity very well, up to 0.5% relative humidity. So in black you see the set levels, and in blue you see the actual humidity here in the insert here. And for the temperature, the same. You see the steps here decrease in the set point, and in red the actual temperature. So we're very closely matching the temperature course here in this test. The same or similar it looks for the CO2. So we can also now change the CO2 content here, varying it between 805 and 827 ppm over the duration of almost 2 hours. And we see deviations of maximum 1 ppm from the set point. Sometimes there are fluctuations due to the feedback between measurement and regulation, mass flow controller, but that remains in the range of 1 ppm. Now, a very delicate point, that is this pressure control of the plant chamber. I said earlier that the plant chamber is connected gas tight to the lysimeter. In order to avoid any problems with air intrusion, we keep the plant chamber at an overpressure of 4 millibar. And this 4 millibar overpressure is maintained with a precision of 0.04 millibar, which means 4 Pascal. And this is a really very high precision control of the overpressure here. You see the set point is 4 millibar and the deviations are maximum 0.04, so 1% deviation plus minus. The problem would be if the deviations were higher, we would have a very strong effect on the weighing of the lysimeters because, well, 1 Pascal is 1 Newton per square meter, and 1 Newton per square meter is the equivalent of 102 gram per square meter. And so we have to, if we want to have a precision of the weighing of at less than 100 gram, then we have to stay below that level. But we managed with a high precision control system of a high precision differential pressure transducer and high precision regulation to maintain this pressure with that precision, so that we can maintain the weighing precision below or with maximum deviation of 100 gram. Without this plant chamber on top, the precision would be even 1 order of magnitude better, plus minus 10 gram. But well, we decided to go for that, let's say, overpressure approach to have always the security that no ambient air intrudes into the plant chamber, but whenever there's a leak, that air exits the plant chamber and it does not mix with ambient air. Now to the plant chamber lighting with an 8-channel LED array. Our aim was to mimic the solar spectrum as much as possible. And in the initial talks with the companies, they said, well, I mean, why do you want to waste energy for the green light? Because the plants cannot use the green light for photosynthesis. That's the reason why they are green, because they reflect this. But we said we want to have, let's say, we do not want to introduce artificial conditions by just having, let's say, the typical artificial light that you nowadays see in greenhouses for growing plants, which is a mixture of blue and red, because these are the two major wavelength regions for photosynthesis. But we also invested quite some, let's say, effort to mimic also the intermediate parts of the spectrum. And with these 8 channels, we now get quite close to, you see in red here now the spectrum of our lighting system, which mimics quite well now the solar spectrum. There are some, of course, some deviations, but that's hard to avoid. For this, you would need even more channels. And then it's sometimes hard to get LEDs with a specific wavelength. For example, here in this region of 670 nanometers, some LEDs are either not available or then have only a short lifetime and so on. So you have to accept some compromises, but we were quite satisfied to be very close to the natural sunlight. The lysimeters are hanging in the basement. They are attached to the ceiling with a steel construction here now hidden under this fire protection layer. And they sit on 3 feet, and each foot carries a weighing cell. So basically the weight of about 3 tons is weighed by 3 weighing cells. And it's hard or impossible to see, but it basically the whole weight rests on 3 spots of less than a square centimeter. And that's the weighing cell. And the weights of the 3 cells are added and then give the total weight of the lysimeter. And as I said, even if you place well, a bar of chocolate on the lysimeter, you will see it in the weight. So we achieve now a precision of 50 to 100 gram. As I said, in the field outside, the precision is even higher without this overpressure system in the plant chamber. The side walls and bottom are insulated. We want to have a very realistic temperature profile. So what is usually neglected in greenhouse experiments is that the soil compartment, the pots are not insulated and get unrealistically hot in a greenhouse. And so we spend quite an effort here in insulating the lysimeters. The current version, this is the prototype. The current version looks now black because we changed the material to Amorflex and now can control the bottom here, cool it or heat it if we want. And so that the temperature profile of the soil is only now governed by the upper and lower boundary, not by any heat exchange between the side walls and the ambient air. We can also control the lower boundary hydrologic conditions there's a suction cup array in it. So whenever the conditions are towards leaching or seepage water, then we have this collection of seepage water in this tank here, which is also weighed. But if conditions like in summer with a strong evaporative demand or strong uptake of water by roots, then we can also pump water back to the bottom of the lysimeter, so to also have very realistic hydrological conditions. The temperature, moisture, and tension, water tension are measured at 6 different depths. And we also have, in addition, suction cups at these 6 different depths, so that we can also analyze soil solution at the different depths, but also then the chemical composition of the water in the seepage water tank. Then outside, where we have that cooling units, you've seen it on the schematic drawing already, air supply unit here, and then redundant cooling units for the plant chamber and the lysimeters. So the question finally now is, what do we do now with these 6 units? 8 would have been better, of course, but a matter of cost and space. We have now 6 units. The question is, what do we do? Do we replicate now 1 or 2 treatments and then get a replicated design? And well, by this will be perhaps able or would be able to measure differences of 2 treatments more precisely, but we don't see, would not be able to see any trajectories or how the changes take place. Is it a linear change or non-linear change or various possible non-linear changes? So the other approach is a gradient design, by which we would then suppose the 6 units to different scenarios or different conditions, and then cover a gradient, and by this would be able to see the non-linear changes much better, but of course without replication then. So in this paper, they looked intensively at the pros and cons of replication and not replication, but gradient designs. So if you replicate, then you have the chance to see a difference in the numeric response among treatment groups more precisely, of course. So you can quantify the effect size of treatments very well by this, but it's bad for detecting non-linearities or tipping points. Basically, if you have only 2 treatments, then it's impossible to decide whether this change was now linear or non-linear, or whether you have passed the tipping point between these 2 states. A gradient approach that allows now allows for detecting non-linearities or tipping points, but the drawback, of course, is that you cannot reliably quantify the effect size of the treatments because you do not have the replicates at the different levels, basically. But we decided to go for a gradient design to make, let's say, the best use of this facility, to go for a gradient design. And in this paper, they showed that even without replication, you can get a good R-square, low skewness, and low kurtosis without replication. If you look at the blue line here, which represents the non-replicated design here in a soil respiration experiment. And so therefore we decided that we think it's scientifically sound and also safe to go for, or even better to go for a gradient design than for a replicated design, which would only allow two treatments, to compare two treatments, like let's say nowadays climate with one future climatic condition. But then the question is, what do you combine? Which CO2 level, which temperature, which moisture, which drought, or which precipitation scenario? And that would be kind of arbitrary choice then. Colleagues in Hasselt, so this is the paper I already mentioned in the beginning, they go for a very similar gradient design. They have 12 ecotrons, similar to our approach, but with the big difference that they do not have artificial lighting, but they rely on solar radiation. So each of those 12 ecotrons is subject to the same light conditions, no matter what you have intent or what you had in mind. You are always subject to the everyday or day-to-day variation of solar radiation. But they have similar gradient design here with 5 different temperature levels and with the corresponding CO2 levels that lead to that temperature change, and also the corresponding precipitation change. And that's very similar to what we have in mind, what we are going to do. So that's let's say these are now that's basically the summary of the experimental design of the first planned experiment. We will take soil from Merzenhausen, which is close to Jülich, in fall 2024. We will then run it for several years with a common crop rotation and management system, and will implement 4 different climate change levels: +1, +2, +3, +4 degrees. The +1 is the current day the current climate, because in the climate change community, the +1 is already what we have now. We will skip the ±0 because that is it corresponds to early or mid-20th century climate. We will skip. The most extreme scenario, +4, we will combine with and without irrigation, and also intend to have a drought and heat adapted genetically modified wheat variety. Then we choose 5 recent years, including the extreme years 2018 and 2021, so a drought year and a wet year, as a baseline, and then subject this period of 5 years to different climate storylines corresponding to a +2, 3, and 4 degree scenario, and then statistically downscale these storylines to the Merzenhausen grid cell, and use either linear interpolation of 3-hour on-scale data to calculate 10-minute weather data for controlling these hour units. So with this, I'm at the end. I have special thanks to our colleagues from our central engineering department, especially Joschka Neumann, who is the central person there and has done a tremendous job in keeping everything together and planning everything, but also other colleagues like Patrick Chaumet, Jan Hoover, Peter Kirchner, Gallup Natur, he is the head of this department, and many more colleagues from there. Special thanks also to our IBG3 team, Norman Hermes, who also does a great job, Thomas Bötz-Halifaken, and also many more who are involved in this. And then, of course, you for your attention.